Hello dear students, we are studying anoxygenic photosynthesis mechanism. So we have discussed some examples of anoxygenic photosynthesis. In our last lecture, we have shown about the purple bacteria, the electron flow in the purple bacteria. In this lecture, we will see about the green bacteria, how electrons flow in the green bacteria. And this all are light reactions as they require the light for excitation of electrons. So this all up till now what we have studied is all the process carried out under the light reaction. Okay, they need the light, hence they are termed as the light reaction. Green bacteria, these are some species are green in color and some are brownish in color. Okay, some are green and some are brownish in color. So though it is green, but it is a bacteria, not a plant. Okay, so hence it is known as the photosynthetic bacteria. So some species are green in color, some species are brown in color. Green in color because of presence of pigment called as bacteriochlorophyll and bacteriophyophytin. These two pigments provide the bacteria green color. Okay, and some species are brown in color due to the presence of carotenoids. These are also the pigments, same like that of the chlorophyll and stuff. Okay, so these carotenoids are also the pigment. These are gram negative. They contain the bacteriochlorophyll C, D and E plus small amount of bacteriochlorophyll A. So these are the pigments which are present in the green bacteria. These are the photosynthetic bacteria which perform the photosynthesis in the chlorosomes. Okay, they perform the photosynthesis in the chlorosomes, and that is spread in cell membrane. Okay, so these are the photosynthetic apparatus present in the green bacteria is chlorosomes. They do not require any type of vitamins for their growth. And this green bacteria is further divided into two types, like same purple bacteria. How we saw that in purple bacteria, purple sulfur bacteria were there, purple non-sulfur bacteria was there. So same green bacteria is divided into green sulfur bacteria and green non-sulfur bacteria. If we'll speak about the green sulfur bacteria, they belong to the family TYBSc. These are non-motile, rod-shaped, spiral-shaped and cocci-shaped. Chlorosomes are present in the cell. They are strictly anaerobic and obligate phototroph. So these are strictly anaerobic organisms. As we are seeing the anoxygenic photosynthesis mechanism, so there is no any involvement or evolvement of oxygen. Okay, no involvement plus no evolvement. अंदर भी नहीं और बाहर भी नहीं. Okay, so there is no involvement and evolvement of oxygen, hence it comes under the anoxygenic photosynthesis. So all we are seeing purple bacteria, green bacteria, these are the examples of anoxygenic photosynthesis. Okay, so this they deposit sulfur extracellularly. Examples of green sulfur bacteria are chlorobium, periodicyon, chloroherpeton. These are the examples of green sulfur bacteria. If we speak about the green non-sulfur bacteria, these are filamentous, gliding bacteria and thermophilic in nature. So this bacteria, they are motile. Okay, green non-sulfur bacteria, they are motile and the movement is done with the help of gliding action means slippery. Okay, sleeping like action, slippery like action, that is known as the gliding, and these are thermophilic in nature. Okay, the pigments included in green non-sulfur bacteria are bacteriochlorophyll A, C and carotenes. Carotenes are also pigments. Okay, Chlorosomes are present when grown anaerobically. So this chlorosomes, this are the, here also we have shown the word chlorosomes, which is the photosynthetic apparatus in green bacteria. So it is present in both sulfur as well as green non-sulfur bacteria. They are photoheterotropic and photoautotropic, which show gliding movement. Means they are motile and movement is done by the gliding movement. They do not deposit sulfur, hence they are known as the non-sulfur bacteria. Example is chloroflexus. So this is the diagram of green bacteria. This green round shape, circle shape are the pigments, bacteriochlorophyll pigments are there. Light is been bombarded. These are the pigments, and the curve type which is shown, it is the chlorosomes. Okay, that is the photosynthetic apparatus. This is the membrane where in this diagram actually our photosynthesis process in green bacteria is been carried out. That is all about. Now here comes the actual mechanism how electrons flow in the green bacteria. First of all, it is the light reaction. Okay, so it takes place in the presence of light. If we'll see the flow of electron, then as we have shown in our previous lecture that light is been harvested through this antenna pigment. Same mechanism is there, just there is a change in the components, and rest mechanism is same, that there is flow of electron from ground state to excited state and again back to the ground state. This movement, which movement? This movement means from ground to excited state, from excited to ground state. This movement, it creates the pumping of H plus ion, okay, and which ultimately leads to the production of ATP. That is just a concept behind all this mechanism. So here also antenna pigments are there. This antenna pigment, they harvest the light, they capture the light and pass it to the reaction center. Here reaction center is photosystem 840. Okay, photosystem 840, or it is also called as the bacteriochlorophyll 840, or you can simply say in some diagram it is P840. You can write P840 as well as you can write this bacteriochlorophyll 840. It's one and same. Okay, the meaning is that it absorbs the light at 840 nanometer. So this is the reaction center or photosystem. Okay, so first step is common. Light is being harvested by the antenna pigment. This antenna pigment harvest or capture the light and transport it to the reaction center. Your reaction center is P840 or bacteriochlorophyll 840. As light is being entered into the reaction center, from ground state it become excited, it reaches to the excited state. Okay, so light falls at this P840, it excites 840 P840, and after excitation what happened? It is rich in electron. Okay, and this scale that is reduction potential scale is same for all the unoxygenic mechanism. Okay, so this it reaches to the bacteriochlorophyll 840 in the excited state. From this excited state, the electrons are transferred to bacteriochlorophyll 633 complex. From bacteriochlorophyll 633 complex, it reaches the iron sulfur cluster protein molecule. It is the protein molecule. Okay, iron sulfur cluster. From iron sulfur cluster, it reaches to the quinone molecule. Okay, the difference between, if we will see the difference between purple and green bacteria, here first quinone pool is there. Okay, quinone set is there, but in green sulfur bacteria there is absence of this quinone pool. Okay, quinone pool is not there, it has been present here. just one quinone molecule is there, but in purple bacteria there are sets of quinone like quinone A, B, and then quinone pool. Okay, so here iron sulfur cluster protein is there. From iron sulfur cluster protein, the electrons entered or passes to quinone molecule. Simultaneously, here ferredoxin play a very important role. Why? Because it reduces this NAD into NADH, and this NADH, it work as a energy storage form, or it work same as that of the ATP. Okay, so here ferredoxin does all the job. It can reduce this NADH. It can reduce this NADH because it have high reduction potential. Okay, it is fully rich protein. It is fully rich protein, so it can reduce this NAD into NADH. Okay, so here ferredoxin plays a very important role. So from this iron sulfur cluster, electrons get transported to the quinone, from quinone to cytochrome BC1 complex. From cytochrome BC1 complex, it enter into the cytochrome C 553 complex, and then back to the ground state. Energy becomes low. Okay, as it is the negative value. See the value here, it plays a very important role. Okay, from negative to positive. Positive, it indicates that it can accept the electron. It doesn't have electron, so it accepts the electron from the other source. Okay, and here the source is hydrogen sulfide. Okay, the source here is hydrogen sulfide. So here hydrogen sulfide, now it is the positive value. Okay, means about 0 and positive means what? It is becoming deficient in electrons. It is becoming deficient, so it requires the electron. Okay, so these electrons are provided by this hydrogen sulfide to this cytochrome C553, and at the same time this hydrogen sulfide get converted into the, or get reduced into the sulfur. Okay, so or get converted into the sulfur, and then back electron enter into this our photosystem P840. So this is all about the flow of electrons in green bacteria. So here H2S is providing electron. Okay, H2S is providing electron to reduce NAD plus into NADH. Even ferredoxin is sufficient. Ferredoxin is sufficient to reduce this NAD into NADH, plus this hydrogen sulfide is also providing the electrons to reduce NAD into NADH. Okay, and then it is converted into the sulfate ion. H2S get reduced into the sulfate ion, and then here it is it reaches back to the ground state because of transport of the electrons. So that is about the all flow of electrons in green bacteria. It is very simple, and the process of the electron flow in green sulfur bacteria and green non-sulfur bacteria is similar. Okay, so there is no any type of difference. Now next is heliobacteria. Heliobacteria, we will see this. They can produce the endospores and grow up to 42 degrees Celsius. These are green in color, and they are found in the paddy fields, means at the rice fields we can say. They contain the bacteriochlorophyll G. These are gram positive, motile. Motility is by gliding or by means of flagella. Okay, and the shape is rod shape. So it shows movement by flagella or gliding movement. Gliding is nothing but it is like a slippery movement. Okay, it can slip. So that is the gliding movement. If we see the example include the Heliobacterium and Heliobacillus. These are the example of this Heliobacteria. So this was all about this different types of the bacteria under an oxygenic photosynthesis. In next lecture, we will study the comparative account of purple bacteria, green bacteria, and even this Heliobacteria. Okay, so our next video will be in the summarized form. and how electrons flow in this three types of the bacteria, that we will see in our further lecture. Thank you.